This is the third part of this kind of session. Now we are going to discuss about what we call sort of a brain framework. So now we are going to talk about how we can learn about different parts of the brain and having a sense about different parts of the brain and how different parts of the brain are involved in cognition. So that is going to be the next step of our discussion in terms of having a neural framework about the brain and how the brain is functioning. And then at the end, we need to be able to make the cognitive frameworks that we discussed about today to the neural frameworks and see how we can have a combination of a neural and a cognitive model. But we need to, first of all, having a sense about what is happening inside the brain. And to be able to have a sense about the brain structure, we need to think about what we call connection in the brain. Because brain is not about independent activities. Brain is about how different parts are interacting. And none of the brain functions would be happening alone without any connection with the other parts of the brain. So brain is an hyper-connected organ. And we will discuss about different dimensions of connectivity in the next sessions. But today I'm going to give you a few examples or a sense about what is connectivity and when we talk about connectivity in the brain. When we talk about foundations of connectivity, we are talking about basically one basic principle. When we have two neurons or two areas in the brain that are working together or oscillating together or fired together or activated together, these two areas are connected together. That is the way that we define connectivity in the brain. And connectivity could be investigated by just the structural fibers between areas, or it could be just based on how the functions of two specific areas in the brain are related to each other, and then they are connected as well. And when we talk about connectivity, there are many different measures to see how two parts of the brain are connected. Okay? We can have structural measures of connectivity, and one of the measures is what we call DTI, diffusion tensor imaging. And DTI is going to be something that we'll discuss in the third and fourth session in details, how that is going to work. And then we will also talk about functional connectivity. When we talk structural connectivity, we are talking about fibers. So we see white matter tracks and fibers that are connecting area A to area B. That is the way that we are kind of measuring the structural connectivity. It is based on the white matter tracks that are connecting areas together, okay? But when we talk about functional connectivity, is about how brains function in one area and function in the other area are kind of related to each other in a way that we need these areas are connected. And based on the way that we are getting signal from the brain, whether we are getting signal from the EEG or fMRI method, then the way that we are measuring this connectivity would be totally different. But I will discuss about some of those examples just to give you a sense about the connectivity. And during the entire 14 sessions, we are going to discuss about different ways of measuring the connectivity in details. Just one few examples. That we know that some areas in the brain are highly connected to each other with white matter tracts. For example, on the left side, you can see the areas that we have here that are mainly, as you can see, these are the posterior parts of the brain. So these are the occipital area. This is the back of the brain. And these tracts, these are basically white matter tracts. Or this is the method that we called DTI before. These white matter tracts are connecting different visual areas in the back of the brain. Or these are basically what we call the motor and sensory areas of the brain. And these sensory motor areas are highly connected with some fibers together. And then we have here some frontal areas of the brain. So if I can just... Okay, so in these kind of frontal areas of the brain, okay, if you see my kind of fibers, these are the fibers that are connecting areas that are in the prefrontal areas. This part is called dorsolateral prefrontal cortex. So we will discuss about these specific brain areas in the next step. But these areas are kind of connected to each other. And while we measure the, let's say, frequency of activities in these areas, we can see that areas in the occipital area are mainly connected within the alpha frequency band, within the beta frequency band, and within the gamma frequency band. Okay? So alpha frequency band for the visual areas, beta frequency band for the sensory motor areas, and gamma band for executive function in dorsolateral prefrontal cortex. But this is good to know, but there are many more details about that. How these DTI images are related to these, let's say, frequency bands, and what do they mean, and what are the details about these, let's say, beta frequency band. Is there any chance that we can modulate these kind of frequency bands and make areas connected to each other? These are the discussions that we will talk about in details in the next steps. But these are the things that we are talking about, how different parts of the brain are connected to each other with, let's say, based on the white matter tracks, based on the frequency of oscillation. So there are different ways of measuring that. But to be able to have what we call kind of the connectogram, what we have been discussing in the kind of previous step, discussing about what are the areas that are connected to each other. The first kind of step would be having an idea about what are the areas that we have in the brain. And talking about the areas in the brain, one of the ways to what we call brain parcellation. Brain parcellation is how we divide the brain into some areas. Some, not just frontal, occipital, parietal, temporal. It is just, the brain lobes are really, really large. We are talking about the subregions within these lobes. How we divide them. One of the best ways that people have tried to use is what we call a cytoarchitectural way. So it's about how brain cortex is kind of organized, its kind of cellular architecture. And a really brilliant guy, it's a German neurologist, Corbinian Brodmann. He was able to divide the brain based on the cytoarchitectonic map to 52 areas. So we know that through the entire cortex, we have 6 layers of cells that are totally distinguishable, even when you see them under the light microscopy. These 6 areas, these 6 layers are different in different parts of the cortex. And based on the differences in their shapes, we can have different, what we call cytoarchitectonic maps. So he was able to divide these areas based on these kind of cytoarchitectonic kind of features. And as you can see here, based on these kind of cytoarchitectonic features, he was naming them as something like area 19, area 17, area 4. And this is the actual map. So dividing different parts of the brain to, let's say, area 10. So area 10 is in the, as you can see here. The frontopolar area that I told you is basically a part of area 10. And then area 9, 8, 6, 4. So the 4 area is the primary motor cortex. And then we have other areas. As you can see, area 12 and 11 are a part of the brain that we call ventromedial prefrontal cortex. Or the areas that you have here, area 44, 45, 47, they are a part of the brain that we call inferior frontal gyrus. So you can see different areas of the brain, they have different, let's say, Brodmann areas on them. And that is one way of, let's say, dividing the brain into some areas. Okay, and he has done a really nice job. Even right now, we are still using the Brodmann areas. It's a really well designed map, and we are using that in the human brain mapping right now as well. But there are some other ways of doing the mapping. There is another atlas that is called the Zero Atlas, the Zitro Atlas. It has been done by a group of people in France in collaboration with a group of people in Harvard University. They have divided the brain into 74, each kind of lobe of the brain, right and left, to 74 areas. Okay? And these areas are basically made based on, as you can see here, based on the gyri that we have in the brain. So it's based on the gyri and sulci that we have over the brain. So these protrusions, they're labeled as one, let's say, area, and then this gyrus, another one. So they try to kind of divide them based on the mainly anatomical landmarks. So it is not about their function. This is not about their the things that they are doing or their cytoarchitectures. It's just about their basic anatomy. And so they divided, as you can see, the lines are based on the sulci. So each sulci would make a boundary for a specific area. So here you can see different areas are divided based on the sulci. So that atlas is kind of based on what we call kind of the anatomical atlas, based on just the anatomical landmarks. So they made the brain into 74 areas. So this is one of the atlases that people use mainly when they use the softwares that are about brain structure, not about brain function. Okay? But we are going to go for the next steps. How can we use these atlases to have a better idea about how different parts of the brain are connected and are working together? One interesting thing that people are able to do with these atlases, because when you have these gyri and sulci, it's really hard to see what is happening under a sulci, which is going to the deep to the brain, and see what are the things that are happening. What they can do in something like a computational way, they can just inflate the brain and make an inflated brain. And the inflated brains would make things much easier to see, whether they are within the sulci or on the top of the gyri. So that would make things much easier to see. So what you see here, for example, here we have the insular cortex underneath this sulci, but here you can see that specific part of the brain, the insular cortex, would be out here. So you can see here the insular cortex that is inflated in a way that you can see it out of the sulci. So you can see what are the things that are happening in what we call the inflated parcellation. So that is another way of kind of visualizing what is happening, what is happening in terms of the anatomy in the brain. When we use these atlases, the anatomical atlas, the other thing that we can do, we can separate the gray matter from the white matter, and we can computationally measure the thickness of the cortex in all these areas. So we can make an average in terms of the areas that we have in this specific kind of area in the brain, and measure the thickness that we have. And not only the thickness, but we can also have, so when we have the thickness and then we have the surface area, obviously based on these two measures, we can have the volumes. So we can measure the volume as well. And here we can also measure the level of curvature that they have, if they are pretty much flat or they are curved. So that is another thing that we can measure, whether they are flat or curved. So there are different measures that we can have for each of these, let's say, brain parcels. So these are specific regions or different parcels in these atlases. And we can measure all these, let's say, structural measure from those areas. And beyond that, we can visualize them like this. What does it mean? It seems really complex in the first look, But that is something that you are going to learn in details, how this map, this is another kind of level of visualization for the entire brain. So those 74 areas that I told you, they are depicted here in kind of abbreviated terms, from frontal cortex, insular cortex, limbic areas, parietal cortex, occipital cortex, and subcortical areas. And then you can see here, these are the kind of the areas that are labeled as the limbic. When they talk about limbic areas, limbic area here means mainly the cingulate gyrus. I prefer to name it as paralimbic, but I mean, that's another discussion. But here they are naming that as limbic areas. So each of these areas are related to those, let's say, 74. Here you have more than 74 because there are some revisions in the atlas. So people have different versions from something like 74 to something like 79 or 78. There are different versions. You can count. I'm not sure what is the version that they are using right now, but you can count the numbers of the areas that you have here. And here there are some subcortical areas. If you know the name, here is the putamen, pallidum, caudate, nucleus accumbens, amygdala, thalamus, and hippocampus. And also this part is the cerebellum part, cerebellum. And this one is brain stem. Okay? So obviously they have simplified that, because when we talk about cerebellum by itself has many areas. So we know that's just too much simplified. Even when we talk about brain stem, it's, such a complex organ. But this is really a simplified way of showing different parts of frontal, insular, limbic. And what do these color means? I mean, these colors that you are seeing here, for example, these colors, blue, greenish, can you see them here? These blue, greenish areas? Elahe, could you see them? I can't see your video right now, so you can just shake your head. I can see that you are seeing them or not. Do you see them? Yes. Okay, Elahe. Okay, good. Okay, so if I go back to the figure that I showed you before, I told you these are the insular areas. Could you remember that we had? Let me just kind of erase the. Okay, so can you see these blue greenish areas here? These blue greenish areas are the insular cortex. And if I go back to what we had here, I mean, these are the blue greenish. So these color codes are related to the maps that I showed you before. So these kind of the outer circle is about the color codes that I showed you before. And then the inner circles would be about the volume, the surface area, the cortical thickness, the curve. So these are the characteristics of each individual region in that atlas, in that cortical atlas. It's not just cortical, it also has subcortical areas here as well. So you can see these subcortical areas underneath. If I would just get zoomed into what you can see here, okay, so these are the parts of the frontal areas. Do we have anybody that would be able to, read the items that we have here? Would know about kind of probably what does it mean to have kind of MFS or some other parts? Let me just, I do not have access to this one. Let me just try to, okay, so let me just bring this. That might make things a little bit easier. Okay. So I provided here some few examples of, for example, this one is inferior. Oh, let me just go back. Okay. So this one is inferior frontal gyrus opercular part. Inferior frontal gyrus orbital part. Inferior frontal gyrus triangular part. And these are three major areas in the inferior frontal gyrus. And those who kind of know what we are doing in the field of kind of language, control, consciousness, This area is critically important, inferior frontal. I love this area. It's just one of the areas that when it gets activated in the patients, I have a really good sense about what is happening in the brain when we have activation in these parts of the brain. This part is the inferior frontal gyrus. And inferior means in the lower part. It is in the frontal part. It's the gyrus. So it's an inferior frontal gyrus. We have pars opercularis, means the opercular part. Pars triangularis, it means it's a triangle here. And pars orbitalis, because you have eyes here. That is the place that we have eye, okay? So this is the pars orbitalis. So we have the pars orbitalis, opercular, and this one is inferior frontal sulcus. So that is the inferior frontal. So that is the part that you do not see here, but it goes deep into the inferior frontal sulcus, okay? So that is the part that goes into that. So you can see these areas are labeled here, and so you can find these labels here, and you can relate them to the anatomy of the brain. So this is how the anatomy of the brain and these areas are related, and then you need to know about these different areas. But now we need to go one step ahead and think about how these areas are connected. Let me just go to the end. That is how these areas are connected. So now we go to another level and not only show these areas around, but also show how these areas are connected to each other with white matter tracks. Okay, so this is what we call connectogram. And when we talk about connectogram, it's not only about brain areas or brain kind of functions independently, but also how these areas are connected to each other. Okay, it's quite interesting that there are specific areas in the brain that you can see that the right and left are highly connected to each other. Most of the areas on the right and left are highly connected to each other. But there are also areas that are within, because this one is the left part, this one is the right part. So this one hemisphere, this is the another hemisphere. So there are areas you can see here. For example, this area is highly connected to another area from right to left. So you see that areas that are connected on the left to right. But meanwhile, there are areas that are connected from within the one lobe. So how these areas are connected. This is one measure of connectivity, okay? And this could be for one single patient. And then I can measure the same connectivity map or connectogram for the next patient and show, okay, if I have another patient, what are the differences? And how these differences are going to inform me about what are the challenges that this patient is facing. So that is an interesting part about cognitive neuroscience. But we are just in the first initial step. So we'll still need to learn many more details about that. But that is the prospect of this course. At the end, at least you would be able to learn what is happening in these connectogram maps and the details that we have in these connectogram maps. These specific figures that I'm showing you are from an article. It's a joint collaboration between UCLA and Harvard University. It's about mapping a patient with brain injury. Here I just showed you the normal maps. Tomorrow, the next week, I will show you the patient with brain injury and how they are different from each other. So it is another step that we'll discuss next week about. But it's not something about kind of the things that we're going to talk about today. But if you are interested to go to the details about the maps that I showed you, this is the paper that you can go and read and find the details inside that paper. Okay, so let me just get you to another level. Till now, I talked about two atlases. One was about brainotome, it was about what we call Brodmann areas, and then we discussed about the Desidero atlas. Now we are going to talk about another atlas that is called brainotome atlas, okay? That is the end of this part. I'm going to give you some hands-on experiences on using another atlas that considers both anatomical landmarks
and also psychoarchitectonial maps. So they basically integrated them together to be able to have a new atlas. Please go inside your web browsers and just search for Brainotome. Just bring up the Brainotome in your browsers and try to find the atlas. Okay, this is from a really nice group of people in China that are doing a really nice job. I really love what they are doing. Okay, I go to the Brainotome. Please go to your Brainotome and then go to the atlas part. And then open the atlas. Now you have access to the atlas. Okay. Then you can go into the frontal part of the atlas and open different parts. You can open superior frontal, middle frontal gyrus, inferior frontal gyrus. So you can open different parts. Let me just go to the inferior frontal gyrus and open the inferior frontal gyrus. So now you can see here that within the inferior frontal gyrus, you have more areas than we discussed before. So we have area 44, left and right, 45, left and right, 45C, 45R, 45R. So there are, you can see that we have different areas. If you remember, when we have been talking about inferior frontal gyrus, we had area 44, 44, 45, so that we can have different Brodmann areas here. So if I go back to the slides that I have, if I just bring up the Brainotome atlas, if you remember, 44, 45, these two areas are mainly on the inferior frontal gyrus part. If you remember them, this is the inferior frontal gyrus, okay? And this is the, as you can see here, different atlases, different groups of people are using different versions of naming for these areas. That could be a little bit confusing, but you can kind of learn about details, and it's not going to be that much complex. So I can go to all these areas, find those areas in the brain, and also it would give me, let's say, how these areas are involved in different sort of cognitive functions, and what are the experimental paradigms that could activate these areas. So going back to what has been asked in the class in terms of, okay, what is the experimental paradigm that we can quantify activation on those specific areas? Okay, these are the experimental paradigms that would be able to activate kind of this specific area. And other than that, I can measure, I can show the connectogram of that area, okay, and show that, okay, this area, what are the areas that are connected to inferior frontal gyrus and that part of inferior frontal gyrus, okay? Do you see what are these areas and under the connectogram? Could you just type them in your chat space? What are these areas? If you take a look, if I just zoom in these areas, okay. Amygdala, hippocampus, basal ganglia. It's ventral caudate, globus pallidus, nucleus accumbens, ventromedial putamen, dorsal caudate, dorsolateral putamen, medial prefrontal. These are going to be the thalamus part. So I'm not going to discuss about the thalamic part. But as you can see here, these areas are connected, highly connected to that specific area in the brain. Not amygdala very much. Mainly hippocampus, basal ganglia. These areas are highly connected to that area that we have been discussing about. Let me just go to the other areas. Okay, this one, another pattern. There is another area, I mean, this is the left one, this is the right one. You can see here. None of them are very much connected to amygdala. Now, 45. 45, there are some connection to amygdala. Left, right, left. Now we are in 45 RL, which is more connected to amygdala, to two different, both medial and lateral amygdala. I mean, both part, because we've been talking about amygdala, it has two parts, both medial and lateral parts. Both parts are sort of connected to that specific area. So you can see the maps of these areas and how these areas are technically connected to subcortical areas. And you can see the connectogram, how these areas are connected. For example, these areas in the inferior frontal gyrus mainly are connected to one side of the brain. Okay. If I go to the superior frontal gyrus and bring things in the superior frontal areas, one of the interesting things that you can see here, what is interesting here? The first thing that you can see that is interesting here is that these areas are mainly connected to both sides of inferior frontal of subcortical areas. And those who know that how much important that we have hyperconnection between what we call dorsolateral prefrontal cortex to subcortical areas for controlling anger, emotions, impulses, you can see that areas like dorsolateral prefrontal cortex are highly connected to both subcortical areas. But when we talk about inferior frontal gyrus, these areas are mainly connected to the same side of subcortical areas. And why it is important, why it is critical when we design a trial, when we are doing the stimulation on dorsolateral prefrontal cortex with magnetic stimulation or direct current stimulation, that is going to be critical to discuss about. So I will discuss about those things. Why we have different results when we do TMS on the upper part of DLPFC compared to when we are going to the lower parts, and how these parts are different from each other. So these minor details are going to be critically important in terms of how we are modulating, because most of the brain stimulation techniques that we have, the non-invasive brain stimulation techniques that we have, they do not have any access to subcortical area. But we know that these cortical areas and the prefrontal area, they are highly connected to the subcortical area. So if I know that where I'm kind of stimulating in the prefrontal cortex and how these areas are connected to the subcortical areas, then I can plan for accessing to these subcortical areas in a way that I'm looking for and in a way that I'm planning for, to be able to have the maximum clinical impact in the kind of study that I'm going to do. We are still talking about experimental design and clinical studies, not really clinical practice, but they are going to inform our clinical practice in the future. So that is the way that we are going to explore. So this is going to be your homework for the next session. You are going to go into the details of what is happening in the Brainotome Atlas, look in their website, see what they are doing, and you are going to write just one single page report. So it is not going to be 2 pages. It's just going to be one single page. It's up to you to choose the font size, the line space, the layout. It's up to you. You can put 1 million words in one single page, but it's going to be just one single page. So you are going to submit one single page, personal experiences of seeing what is happening in Brainotome, and tell us what are the things that you have learned from exploring what is happening in Brainotome Atlas, and what are the things that they are doing in Brainotome Atlas, and how you are. I'm really interested to see how you're going to organize your report in one page. So it is going to be one page that you are going to submit. That is something that I will be discussing at the end of the class as well. Okay?